So thank you very much for the introduction, and we're excited to be here to talk about some of the next blockbusters in immunology. I'm Cameron Taylor, Managing Director from the BofA Life Sciences team. Extremely delighted to talk about this field where there's quite a bit going on. Obviously, from my perspective, a lot of this is the numbers and the blockbuster nature of these products or these billion-dollar products. We've seen probably about 20 plus products that are multi-billion-dollar products in the field, and it creates a lot of focus from pharma, from investors, and others. But it's much more than just the financial elements. So I want to introduce my co-moderator. So doctor, feel free to take it away with the perhaps more important patient element.
Thank you so much. So I'm Gravallese. I'm a rheumatologist and immunologist and chief of the Division of Rheumatology, Inflammation, and Immunity at the Brigham and Women's Hospital. And of course, in my field, we see chronic diseases, and we know there are a number of unmet needs in these chronic diseases. And we have several companies here who are looking at novel approaches across a variety of indications for immunologic diseases and looking for that next blockbuster medication. And so specifically, these companies are looking at antibodies and modifications to antibodies and modifications to antibody processing, as well as oral agents and oral peptides. And so before we dive into the questions, I wonder if I could ask the panelists to introduce themselves and provide some background to the technologies that you're using and the approaches that your companies are making to the immunology space. And perhaps we could start with you, Tom.
Sure, happy to. And first of all, thanks for hosting us, Cameron and Ellen. It's very, very much appreciated. I'm CEO of Jade Biosciences, Tom Frohlich. We're a relatively new company that was founded in the middle of last year. We were founded on 3 assets that we have access to from actually a company here in Boston called Paragon, who are protein engineers, very talented at developing high affinity bonding monoclonal antibodies, but their particular expertise is in applying half-life extension technology, which is extremely useful for 2 reasons. One, you can get more complete coverage of the target throughout the dosing interval, which should provide better efficacy and clinical activity. And then, of course, with a long dosing interval, it can be much more convenient for patients, and many of these diseases are lifelong chronic therapies, so minimizing that treatment burden is extremely important. Our lead asset is actually targeting anti-APRIL, which the lead indication is IgA nephropathy. We think it's a very large indication, even though it's a rare disease. I know that's a little bit of an oxymoron, but we believe in the US alone, it's a $10 billion plus branded opportunity. And we have strong conviction that the anti-APRILs are going to move to frontline therapy. They have the possibility of being disease modifying. They take away that pathogenic driver of disease, that galactose deficient IgA, and then that translates into large drops in proteinuria, quite often back into the normal range for patients, which ultimately gets them what they want, which is stabilization of kidney function. We believe with the technology that we have, we can create the best in class anti-APRIL because we can get deeper levels of APRIL inhibition, which translated to better activity, but then of course do that in a very convenient way. Patients with IgAN are diagnosed typically in their 20s and 30s and need lifelong treatment, so infrequent dosing will be very, very valuable for them. Very exciting time for Jade and I think the field in general.
Yeah, thanks very much, Tom. Dinesh.
Sure. So the instructions I was given was I should briefly introduce myself and then talk about the company. So I'll do that. So I have been in the sector for close to 40 years, started in the mid-1980s as a medicinal chemist at Bristol Myers Squibb. In the 1990s, I moved to the Bay Area, started working for biotech companies, and the entrepreneur bug in me, it got a lift after being in Silicon Valley. So a couple of scientists, we started Versacor in 1996 with a few $1 million. To make the long story short, in 2005, Pfizer acquired us for $1.9 billion. And in between, I transitioned from being a scientist to sort of more like an executive. And then I did a couple of gigs and transitioned from founder to CEO. And in 2008, I started Protagonist in the US as its CEO, and that's the journey I have been enjoying ever since. So being a scientist by training and being trained in small molecule chemistry, I saw the era of small molecule based drugs being in prominence. Then I saw the era of the big injectable biologics come in vogue. And then as a scientist, I was looking always for a differentiation. So in peptides, which is the ground in between, I found a happy medium. And the whole zeal then was like, hey, what if in a peptide we could combine the oral characteristics of small molecules as well as the amazing specificity and potency of big biologics in a single peptidic entity. So that vision, after lots of ups and downs and going sideways, ultimately led to icotrokinra at Protagonist. By the way, at Protagonist, we are focused exclusively on peptidic drugs. And icotrokinra, that's the oral IL-23 receptor blocker. It's partnered with J&J. It's a partnership that started all the way back in 2017, so it has been a long journey for both companies. But the outcome is excellent, very positive phase 3 data in psoriasis. An NDA was filed in July, so hopefully by middle of next year it will get approved. We also have positive phase 2 data in ulcerative colitis. So we believe that this is a drug that will check the box on all 4 indications for which the injectable antibody drugs are famous for. Whether you look at Skyrizi or Tremfya or Stelara, they have been successful in 4 major indications: psoriasis, psoriatic arthritis, Crohn's disease, as well as ulcerative colitis. And I would say icotrokinra is well on its way for proving itself in all those categories. Right now, the proof what we have is positive phase 3 in psoriasis and positive phase 2 in ulcerative colitis. So I'll stop there.
Thank you so much. Michael.
Great. First of all, Cameron, Ellen, thank you for having us. Really a pleasure to be with all of you today. Mike Quigley, Chief Scientific Officer, Global Head of Research for Sanofi. I had the privilege of joining Sanofi about a year ago at this point in time. My background is I'm a PhD immunologist by training, and I spent my career up until now in pharma, venture, and biotech. So I've seen enough of the breadth of the ecosystem associated with those 3 disciplines to, in theory, bring, and in practice, hopefully bring a sort of new lens to how we do research and development within pharma. And I'm really excited to have joined Sanofi at a really interesting time. And maybe the framing for that as it pertains to our conversation here today is I think we're in an immunology 2.0 era. And if you allow me to sort of use that to sort of detect the present and the future of immunology. We need core immunologic expertise. We have that on stage. You'll hear a bit more about today. We need next generation therapeutics and platforms. You'll hear a bit about that today. I have a personal belief you need scale to bring that benefit to the broadest number of patients possible. And Sanofi's at a really interesting time in rethinking what that means as we think about immunology and inflammatory diseases. And we're shifting that lens to have a broader aperture, one of a more immunoscience in nature. That is to say, sort of understanding of immunologic nodes and pathways and how they pertain to disease, but not with a traditional disease-centric or sort of siloed view of the world, but rather a broader view on how those nodes could be generally applicable and bring benefit to patients across the spectrum of a variety of diseases. Core traditional sort of I&I indications, we'll talk a bit about that today, but more broadly, things like neurodegeneration, retinal diseases, rare diseases, clearly driven by, in large part, an inflammatory component that we believe we can bring to bear the same sort of philosophy that we have within I&I, but with that breadth in hand. And I'd say we may or may not get to it today. Sanofi's really, doubling down from an AI perspective and how it enables the progress of those types of sort of aperture, if you will, in immunoscience lens and broadly from a drug development and discovery sort of perspective, and really evolving as we speak the company to much more of a sort of pure R&D driven AI powered sort of biopharma organization. So excited for the conversation here today.
Thank you. Ken.
Thanks for the invitation. My name is Ken. I'm a physician scientist by training. I left academic medicine back in 2007. And then I was an investor for a few years. And it was interesting to sort of look at the landscape of innovation through that lens. And then in 2010, I made the transition to being an operator. And that's what I've been doing for the last 15 years. It's interesting, actually, my career started off in diagnostics. And so in 2009, 2010, had really identified cell-free DNA or liquid biopsy as an area of promise. And then obviously that has now become standard of care in both prenatal as well as oncology. Started a company, we sold it to Roche, and that product was globally commercialized. Shortly after that, then got interested in looking at this whole metabolic space of non-alcoholic or now metabolic-associated steatohepatitis, NASH or MASH, and so worked there for a few years as CEO. And then in 2020, started a company in the radiopharmaceutical space for oncology. Again, another area that when we started was really, I would say, underappreciated. People were pretty skeptical about it, but you fast forward to it now, and many different pharmaceutical companies actually have a very active effort in radiopharmaceuticals, and that company, which was Raise Bio, was acquired in the early part of 2024. And so coming out of that, was sort of looking across the landscape broadly around, is there anything out there that looks really interesting, where I also felt could be sort of both clinically attractive, but also on the early part of that value creation curve. And really came across this really exciting data in the I&I space where various different investigators had taken an oncology drug product, cell therapy or CAR T, and applied it to autoimmune patients and saw really like unprecedented efficacy. I mean like eye-popping efficacy, something like drug-free remission and cures in highly refractory patients. And there's dozens, if not 100+ companies that have cell therapy products going along that path, but I didn't really think that was going to be scalable as you talk about scalability. And sort of, again, borrowing from what we've learned in oncology, it had this idea that you could take bispecific antibodies and actually accomplish the same result. And so we started Candid Therapeutics really with 2 different clinical stage assets that have been originally developed for oncology and then repurposing that for various different autoimmune diseases. You know, those were both in-licensed from China, and I know that there's a separate China panel on this earlier in the sessions. But yeah, so we're focused on now taking forward what we regard as a clinically validated mechanism, but with a much more scalable, patient and provider-friendly format of a product. And we've got a broad-based effort to sort of understand where B-cell depletion, which are the cells that we deplete, actually is a primary driver of the disease, because it's not going to be in everywhere, but I think through generating clinical data, we'll be able to figure that out.
Okay, well, thank you so much for those introductions and for the overview of what you're all doing in your companies. I think we'd like to dive a little bit deeper now into the technologies and the science of the technologies that you're working on, and also your choice of targets. And so I'd like to start with you, Tom. Tell us a little bit about your choice of APRIL as a target, as opposed perhaps to BAFF or to a combination of APRIL and BAFF, and then also about the technologies that you're using to extend the life of antibodies with this BTE approach.
Yeah, it's a really good question, Ellen, because as you know, selective anti-APRILs and dual APRIL-BAFFs have shown clinical activity in IgAN and have shown benefit for patients. So we were quite thoughtful about how do we select the right target for this particular disease. And IgAN is actually a plasma cell-driven disease, and APRIL's really pathogenic in producing IgA, or converting cells to IgA-producing cells. And what we've seen actually historically is when you try to block a broader compartment of B cells, there actually is very little activity in IgA nephropathy. CD20 inhibitors had been tried in the past. Actually, selective BAFF inhibitors had been used in IgAN with no impact on proteinuria or even IgA levels. So we were pretty sure that all of the efficacy that was driven in the APRIL-BAFF dual inhibitors was actually being driven by the APRIL component. And actually, when you look at cross-trial comparisons between selective APRILs and the dual APRIL-BAFFs, actually the efficacy is very similar, really suggesting there is no additive effect of blocking that BAFF. And as I mentioned earlier, patients with IgAN are typically diagnosed between the ages of 15 and 30 and need decades of therapy. So we really strongly believe that in this patients who actually have very few comorbidities outside of their kidney disease, that being as specific with your immunomodulation is going to be preferred over the longer term. So we have high conviction that the selective anti-APRILs are going to become the dominant class within IgA. Different for different diseases, like if you're going after something that is more B-cell driven, like Ken was saying, then that BAFF component is interesting. But for IgAN specifically, we think the APRILs. Within the anti-APRILs, we are third in class, so we are chasing. two that are ahead of us. But we do believe there's limitations with those approaches in terms of those molecules actually can't completely suppress APRIL through the entire dosing interval. So they are leaving some efficacy on the table, and that's what we are looking to overcome with the antibody that we're putting into the clinic. It's very high affinity binding, so over 750-fold more potent in terms of an APRIL binder than the lead asset. But also it has this YTE mutation in the FC portion. And what that does is it increases binding affinity at low pH to FcRn, so it promotes recycling back into circulation. So you actually end up with these really long duration of action. And with that, we can get that complete duration suppression of APRIL, which should lead to better clinical activity, but then also do that with very infrequent dosing. We believe we can do it with one dose no more frequently than every 8 weeks, and potentially longer. We'll have to see what our phase 1 study looks like. But really the opportunity for a best in disease profile with this technology built into it.
Yeah, really exciting. Thank you.
And so for Dinesh, I mean, a slightly different question, right? And you mentioned, right, that you wanted to get the best of both worlds from an oral perspective as well as a biologic perspective. You also mentioned some of the other drugs in the class, at least for your lead. But Tremfya and Skyrizi weren't what they are today at the time you decided to choose that as a target. You've also done work in alpha-4 beta-7. So how did you think about your underlying technology and the selection of your target and getting that right into the gut and getting that level of efficacy? So how did you kind of go through the work of other targets and select those as you thought about disrupting the field?
So I think it's an excellent question, and it has been a great journey, and I'll try to put things in perspective. I mean, in all honesty, we were driven by our self-belief about excellence in chemical sciences. So when we started, and this is all the way back in 2009, 2010, nobody was paying attention to peptides. And oral peptides was, or still rather is, that's like the holy grail in the field of peptides. So, we were much younger then and had a little bit of ignorance and arrogance, and we are like, this is exactly what we are going to try to do. So that's how we started. But of course, then it is like if you make the assumption you can create oral peptides, right? It's a big if, but let's say if you can, where would be the biggest differentiation? And that is where now we started paying attention to the actual science that was evolving at that stage. The other thing we concluded was like maybe the application will be best for chronic indications where patients have to take the drugs for the rest of their life. So having the convenience of an oral pill will make a lot of sense. And that is when it's like, in the evening if you watch TV, it's full of ads of these injectable antibody drugs. And so the IBD drugs caught our attention, and Humira was there, but scientifically we said we are not going to work on TNF blockers. Humira is a great drug, but it's kind of toxic, let's say. And the best emerging targets at that time were alpha-4 beta-7 integrin, right? Entyvio is a great drug coming from here, Takeda. By the way, Takeda are our partners also, but on another drug. And then IO23 was an up-and-coming target in our opinion, and it appeared to have the rare combination of both efficacy as well as safety. So those are the two targets we chose to work on, and as you know, and this is the nature of the beast, with alpha-4 beta-7 integrin, we had PN943, we missed the efficacy endpoint, and could the drug have worked? Who knows, but at the end of the day, the conclusion was like, let's put it aside. And IO23 worked like a charm. And as I mentioned before, we ended up with a partnership with J&J all the way back in 2017 when it was just a preclinical asset. So, and our first generation drug, the concept was we are developing oral but gut restricted peptides. So, because we were targeting IBD, and that concept actually didn't pan out that well. But the clue we got is like, now we need to engineer a bit of oral bioavailability in our peptides. So we were able to do that, and the outcome was PN235, which is now icotrokinra, and that's having a joyride as we speak.
All right, thank you. Ken, I'd like to turn to you and talk a little bit about bispecific T-cell engagers. As you mentioned, in our field in immunology, what we've learned is that deep B-cell depletion is really effective. I mean, sort of surprisingly effective, honestly. And many of the trials have been with CAR T-cells, and now we see these bispecific T-cell engagers coming on board. I wonder if you can make a comparison to the CAR Ts and also maybe to other forms of B-cell depletion with rituximab, for example. And then what do you think about the durability of the bispecific T-cell engagers?
Yeah, so I mean, a couple of interesting things you mentioned there. I mean, the first one is I think the entire field was surprised that B-cell and depletion of just the B-cells, which is just part of the immune system, led to this amount of profound efficacy. And there's a lot of these diseases, I think, where conventionally people thought they were T-cell driven or by other things. Again, I think this is just goes back to, at least for me, I just feel like we still don't really understand biology. And at the end of the day, like clinical data will trump like any type of scientific hypotheses. But there have been antibodies, rituximab, obinutuzumab, inebilizumab, those are all approved or soon to be approved to treat these autoimmune diseases. But a T-cell engager or a CAR T sort of takes it to the next level because it's a much more potent mechanism to really eliminate, people use a term called immune reset to really get rid of the B cells and the plasma cells from pretty much like most, if not all, tissue compartments in the body. And there's a lot of different ways people have now tried to pursue this, and I think this has created actually a lot of confusion in the space because there are multiple cell therapy-based approaches. There's auto CAR T, there's allogeneic CAR T, there's natural killer cells that people are also doing and embedding CAR constructs. And then you have in vivo CAR T, not to confuse people. This you give like a intravenous infusion and it's like either a lipid nanoparticle or a virus that transfects your endogenous T cells to reprogram them. And then you have things that are like the T-cell engagers, which are bispecific antibodies. And I would say that what we've seen, and the data is emerging, but it's confusing because everyone's generating data. And this happens in our industry all the time, where I think different snippets of data come out, and then quickly some people may come to conclusions, but the overall mechanism makes sense, but then the question is, what's the right modality? I would say based upon what we've seen, both from academics who are taking approved bispecific antibodies and using them through compassionate use, as well as our own data, is that you are getting the same level of deep B-cell depletion in tissues with the T-cell engager as autologous CAR T. And I don't think that's actually widely appreciated by the field. And it's all contingent upon the dosing strategy, because with the T-cell engager, you got to make sure that you dose it enough to get to that same level of potency. And what we've so far seen in the field is that if you can achieve that in certain diseases, you are seeing patients that are out several years with cellular therapy that are in drug-free remission that had failed pretty much every approved therapy prior to that. And what we're now seeing with the T-cell engagers is earlier because it came onto the scene later, but now we're aware of patients who are more than 18 months out after receiving a bispecific antibody and are actually in disease-free remission as well, drug-free remission. So it's a super exciting time. And again, I think we just need to explore through clinical data to figure out where the B cells really are the primary driver of disease, because it's going to work beautifully in some areas like IgA nephropathy and these other areas where the autoantibodies are the culprits. And then in other diseases where it could be more of a mixed B cell and T cell picture, a pure B cell driven approach to deplete that is not going to be as effective.
Thank you.
And so, Michael, you have a different perspective in that, right, we've had 3 panelists that are biotech companies, underlying technologies. You have an existing mega blockbuster in Dupixent. How do you look at thinking about disruption overall in a particular field? You have, internal assets, you have venture capability, partnerships. How do you look at different technologies, targets to think about this disruption in the field?
Yeah, thanks, Cameron. It's a great question. Maybe I'll frame the answer where we think the unmet need lies today, sort of 3 main areas, and then how we think about different levers we might have available to sort of balance risk, and to Ken's point, sort of outstanding questions, at least in our end or the field's mind, around targets, modalities, et cetera. So for us in I&I diseases sort of generally, on the first hand, clearly, in a sort of pan I&I perspective, there's clearly an efficacy ceiling we need to collectively break through. It differs per indication, but it's clearly there. The increased activity that we need to bring to bear in the next wave of assets is critical. The second aspect of an unmet need perspective is convenience, but not convenience for convenience sake, convenience tied to clinical activity. And so we'll come back to sort of maybe some examples there, whether it's an oral combination type approach or durable sort of long-term sort of extended pharmacodynamics and or dosing intervals. And the third, and Ken hit on it a little bit, is ultimately what we're looking to move towards is true disease modification and sort of ideally treatment-free remission for patients. And so as we think about how we sort of frame our portfolio strategy, it's really in those three areas. A lot of what we sort of do either internally and through partnerships in the efficacy ceiling tends to be through combination approaches, either bi or multi-specific, using our nanobody platform, for instance, or in additional sort of platforms and combination approaches. We also have efforts, both internally and in partnerships, through oral approaches to either targets that have been validated clinically by biologics and or are more novel, perhaps adjacent to those like IL-4 receptor alpha biology, where an oral agent could potentially replace that from an activity perspective. And on the durability sort of part, like Ken, sort of have interest in not only B-cell depletion, but I think other cell subsets of potential interest. I think we're seeing an emergence and a wave of approaches, for instance, in T-cell depletion, and maybe whether or not that will have an analogous potential for immune reset. I think time will tell as those clinical experiments play out. And/or extended pharmacodynamics or off-treatment remission, potentially moving from a current standard of care in atopic dermatitis, for instance, of 26 sort of injections a year to potentially 4 injections a year, as we think about sort of balancing that. So from a portfolio perspective, that's our lens. We don't have a not invented here view. So we use everything at our disposal within Sanofi, either internally through traditional collaborations, Sanofi Ventures as our corporate venture capital arm, and additional investments to be able to sort of sample innovation wherever that might exist.
Okay, thank you. So moving a bit beyond immunology, we'd like to ask you, when you think about your drug development platforms, what are some of the surprising hurdles that you've met along the way? And how have you met these hurdles? How have you approached them? And also, is there advice that you wish you had gotten when you started out that you didn't get?
For me.
Anyone.
I'll start, and my colleague can jump in. I'd say, and it comes back a little bit to scale and where we sit within Sanofi, the breadth of the pipeline. Increasingly, as we've seen, when new benefits brought to patients in areas where perhaps there wasn't an option before, whether that be with the advent of dupilumab in atopic dermatitis, really bringing that safety and activity benefit, sort of being able to move earlier in sort of mild to moderate disease, for instance, and or things like systemic mastocytosis, with Ayvakit and sort of an RTK inhibitor. That's in isolation incredibly important from a benefit perspective. As you start to scale that from a portfolio and a global trial perspective, new standard of care obviously changes the paradigm and the unmet need in these patient populations. And so part of what we knew, but we need to collectively, I think, understand where that unmet need will be in years in the future, is to start to understand the different sort of real-world assessment of, for instance, atopic dermatitis patients, those that have sort of previously been exposed to biologics and or JAK inhibitors. That treatment paradigm will be different, and the disease paradigm is likely different. And so having the forethought to take more of a biomarker precision medicine-like approach to sort of understand subsets of Th2 versus non-Th2 disease, for instance, is something that I think we collectively grapple with and will continue to in a positive light as we make decisions from a global sort of development perspective.
Yeah, great point. Anyone else?
I had 2 very, very quick thoughts. One is it's such an exciting time in immune therapies, and we talk about I&I and kind of B-cell depletion or T-cell depletion as if it's this one broad thing, although we all know it's not. And I think what's really exciting, and I think we need to learn a lot more about, is how do we take the right target for the right indication and get that right level of immunomodulation for that indication with the right benefit risk profile. And as all these different targets emerge, I think we're going to learn more and more about how do we make sure that we're hitting the right B cell compartment or T cell compartment or whatever it is for that particular indication. So I think that's like a really exciting kind of hurdle, but also a challenge. The other thing that's a bit of a challenge as a smaller biotech company is like, as soon as there's a good target, it's a very competitive space. Like everyone sees that these are good. And I think the good advice that we have at Jade is we have to move extremely quickly. We really have to make sure we're putting a lot of resources towards these things, developing them with really strong partnerships with academics and industry and regulators, and just move at pace, because every new target that comes out, there's going to be a dozen competitors, and you need to get out ahead.
Definitely.
I mean, I think for me, the one thing is just that, and I've learned this over the last 15 or so years, is there really is just no replacement for just getting the clinical data and getting the data in any fashion that you can. I think a lot of times, people have hypotheses around this or that, and there's a lot of different things that are being published in the literature around mechanisms or pathways. But I think until you get into the clinic and actually see how a drug or a mechanism is working, you really don't know. And I think our industry as a whole probably spends way too much time hypothesizing and pontificating without just pushing the molecules into the clinic. And I would say that there are mechanisms outside the US, whether through compassionate use or investigator-initiated trials, that are much easier to do. And you can get that initial clinical proof of concept, before you invest significantly into later stage clinical development. And I actually think that model is one that should be embraced and adopted more, because drug development, and people write about this all the time, people say, it costs $500 million, $1 billion plus now to take a molecule all the way through development. And I just think that's incredibly inefficient. And there's no need for that, but the industry is used to that and is wired that way. I'm someone that tries to always maximize on efficiency. And so in all the different instances I've been in the last 15 years, we've always identified ways to generate clinical data in a non-conventional way that every single time has always generated and given us insights that has allowed us to sort of propel forward and like way faster than anyone else in the field at a fraction of the cost. So I mean, I learned that along the way, but I wish I sort of fully had understood that at the get-go. And I think we've been efficient, but there's always ways to push yourself to be better.
Maybe what I'll add is in biotech, of course, most of us come from scientific backgrounds, and scientific excellence is a very dangerous thing, because we are so convinced about being right, and the statistics is most of the time experiments fail, and drug candidates fail. So coming to grips with all that, a practical solution that I found is just focus on differentiation and avoid situations where scientifically you won, but in the end, that the differentiation is not big enough because you're not working in isolation. There is competition out there. A lot of people are working in the same field, and you always have to be sure that you are achieving that differentiation, because you want to avoid winning the battle and losing the war. So that philosophy has really served us very, very well. And of course, Protagonist is at a good spot today, but it hasn't necessarily been easy, and so we stay very grounded. Some of the things we learned is your first drug candidate may not work. Now, as long as you see that differentiation and you learn from the first failure, you can apply it to the second one. Icotrokinra is the third drug candidate that was put in clinical studies. Another thing is your first indication may not work. This is not in the I&I space, but other drug is rusfertide, partnered with Takeda. The first indication we tried, it's a mimetic of the natural hormone hepcidin. and we tried it for beta thalassemia. It worked beautifully preclinically, but it didn't meet the expectations in clinical studies. Second indication was hereditary hemochromatosis. It worked, but the commercial appeal was kind of lacking. And it was the third indication, polycythemia vera, where we found, an excellent outcome. So I think that kind of resilience and tenacity and scientific innovation has to continue all along the line. But, and one way or the other, you have to seek out that differentiation.
So then in terms of seeking out that differentiation, and Ken, you touched upon it, right, there's this cost element as well, right? And each of the companies here, right, has a little bit of a different approach in terms of progressing their assets. Dinesh, you've engaged in a number of partnerships, but how do you think about obtaining the capital and debating whether or not to think about a partnership? And obviously, Michael, maybe your views on how you think companies should view, right, a pharma partnership as well.
Yeah, so I'm a big believer of partnerships, and both of our assets that have found success in phase 3, both are partnered, one with Takeda, one with J&J. And in the early days, partnerships are important because you want to get your approach and the company validated. Partnership will bring in financial resources, but eventually I realized that those are kind of important but not the most important things. The most important thing we get from our partners is the experience and the scale at which they do things. Like if you look at icotrokinra, the number of clinical studies that J&J is pursuing, there is no way a biotech like Protagonist would have been able to do that on its own. In very simple terms, in my own hands, let's say an asset that is fully owned by me, it's a $1 billion pie. There is nothing wrong if by partnering that becomes a $5 billion pie. And even if Protagonist gets 1/3 of that pie, we still come out ahead. So even financially it makes sense. And your probability of success and the scalability that the pharma brings is unmatchable. So I'm a
Ken,
big advocate
I want
for
your
partnerships.
counterpoint. Because
Well, I'm an advocate for partnerships, but at the right terms.
I have a polar opposite view. I'm like anti-partnership, just because in my experience, and I've done partnerships previously with my other companies, is that, I think our objective or biotech's objective is to sort of have a very solid hypothesis and to test it efficiently. And you make decisions very differently than a large pharma partner would. And so in my experience, if you do a partnership, number one, you're going to slow yourself down. And I think number two, you're going to limit sort of the upside if the thing works out. And so I think you limit your degrees of freedom. I would say that if you cannot access capital, then I would look towards partnership. But if capital is available, I would say that I would urge entrepreneurs to sort of hold on to their assets, control their destiny. And there's no reason you can't capture the entirety of the $5 billion or the $10 billion pie if it's working. Because again, there are so few things that have transformative potential in our industry, and we heard from the investor panel that if you're working on something and it's working, that capital will be readily available. And you want to put yourself in a position to either be able to raise that capital and succeed on your own, or you become sort of the must own. And then in that instance, then a wholly owned asset within a pharma, I think, can be incredibly effective. But I think partnerships are incredibly challenging, and I would strongly recommend it.
All right, Michael, you get to prove
I
me wrong.
suppose I'm somewhere in between. I'm somewhere in between.
It's purposely.
Yes, exactly right.
Sorry, I would say just hire a great business development person.
I've never had a BD person in my company.
I think the lens between the two is timing, right? I mean, I think, look, we view partnerships as critical. I stated it already. Innovation comes from within our walls and outside our walls. We view it very much in a risk assessment and sort of gap filling exercise as we think about our own internal portfolio and how we sort of partner. I think what we're trying to evolve to is something maybe more akin, doesn't solve for the timing, but where we think we bring benefit, which is that scale, that experiential sort of aspect of development and weighing in. And we try to be thoughtful about how involved we are on a day-to-day basis versus when we want to be there when you're ready to scale a particular asset or a platform or the like. So I think, that tide rises all ships, and I think our collective, sort of benefit exists in the context of I&I and bringing benefit. And for us, we have a variety of levers that we use simultaneously to try to sort of meet in the middle, depending on different lenses from a biotech perspective.
And now we get the science behind the whole seating arrangement.
There you go. And
well
there we
planned.
have it, folks. A diverse set of opinions. We want to thank all the panelists today and the hosts. So really appreciate the time.
I'd like to personally thank you also for not only for your insights tonight, but for all the work that you're doing in your fields. We benefit so much, and our patients benefit so much from the work that you're doing. So thank you.
Thanks.
Thank you.